Overall, the purpose of this study was to elucidate the interactions between the three proteins by locating the binding sites of 3S on both GC1QR and on C1q. Also, to develop potential methods to inhibit the binding of 3S to receptor GC1QR on CD4-positive T cells in an effort to prevent cell death. All of the following graphs represent ELISA experiments. The x-axes represent proteins or groups, and the y-axes represent optical densities signifying levels of 3S detected. The first step was to identify 3S binding sites on GC1QR, as shown on the graph. As compared to a positive control, it was seen that GC1QR proteins with deletions in residues 144 to 162 and 174 to 180 exhibited significantly lower levels of binding to 3S. These residues were then considered potential 3S binding sites on receptor GC1QR. All deletions were not significantly different from the control and exhibited significantly higher 3S binding as compared to these two residues. Next, the study looked at the first method for inhibiting 3S binding to cells, the peptide-based inhibition. Incubation of 3S and GC1QR peptides were conducted prior to binding to cells with GC1QR concentrations ranging from 50 to 200 micrograms per milliliter. It was seen that only peptides covering residues previously identified as potential binding sites, 144 to 162 and 174 to 180, led to lower levels of 3S binding to cells. This also only occurred at 50 and 100 micrograms per milliliter. Higher concentrations of all peptides caused no significant difference of 3S binding as compared to a positive control. The study also looked at a second method of inhibiting 3S binding to cells, the antibody-based inhibition. An anti-GC1QR antibody, as shown by the two green bars, was added to cells before exposure to 3S. The darker bar represents a higher concentration of anti-GC1QR antibody. As you can see, this method was not only effective as compared to a positive control shown on the left, but was even more effective than peptide-based inhibition shown by the two blue bars on the right, yielding lower levels of 3S binding to cells. Also, this inhibition is dose-dependent, with a higher concentration of antibody yielding lower levels of detected 3S bound to cells. The next part of the study involved the interactions between 3S and C1q. The study looked at the 3S binding sites on C1q. Using C1q peptides, it was seen that 3S likely binds to the A chain on C1q, residues 155 to 164 and 14 to 26, with these peptides yielding higher levels of detected 3S as compared to a negative control, and no significant difference as compared to a positive control. This data supports the work of Thielens of 2003, who stated that there are two areas on C1q that 3S may bind to, one located on the collagen tail, which is represented by residues 14 and 26, and the other located on the globular heads of C1q molecule, represented by residues 155 to 164.